Experiment number 4, qualitative analysis of aqueous solutions. In this experiment, our goal is to use qualitative chemical tests to determine what metal ions are present in a clear aqueous solution. What's the difference between a qualitative and a quantitative analysis? In a qualitative analysis, we're just looking to confirm the presence and identity of a chemical species. For example, we need to answer this question: is lead 2+ ion present in the unknown sample? In a quantitative analysis, we also need to determine the amount of the chemical species. For example, we need to answer this question: what is the amount or the concentration of lead 2+ in the unknown sample? In this experiment, each group will get one unknown solution. This unknown solution may contain one or more of the following metal ions: lead 2+, silver plus, iron 3+, barium 2+. We need to separate these canions one by one, and then we will do a test to identify this ion. It's called a spot test. So here's the flow chart. We'll talk about this flow chart. First, let's assume we get a mixture of 4 metal ions: lead, silver, iron, and barium. The first step is to add chloride, Ni, into this mixture. This is because lead and silver will react with chloride to form precipitate. Iron and barium will stay in the solution. This way we can separate silver and lead from iron and barium. Again, iron and barium will stay in the liquid layer. Lead and silver will precipitate out of the solution. Now, how do we know if we have lead or silver or both in the mixture? Well, it's very simple. We just heat it up. This is because lead chloride is soluble in hot aqueous solution. Silver chloride is still insoluble in a hot aqueous solution. So after we heat it up, if you still see white precipitate in the test tube, then we have silver plus in the original mixture. If everything dissolves, then we know there's only lead 2+ in the original mixture. Alright, so very simple. We just heat it up. Alright, if you still see white precipitate, then there is silver plus. If you don't see any white precipitate or precipitate dissolves in hot water, then there's only lead 2+. Now we need to confirm the presence of lead 2+, alright, in the liquid layer. We need to do a spot test. I'll talk about this later. So get back to here. After we add chloride Ni into the mixture, again, this iron 3+ and barium 2+ will stay in the liquid layer. We need to separate this two, right? So we add ammonium hydroxide. This is because this iron 3+ reacts with ammonium hydroxide and form a precipitate. Barium 2+ will stay in the solution. So this is how we separate barium from iron. So again, in the liquid layer, you have barium 2+. And then we do a spot test for barium 2+. And after we precipitate this iron out of the solution, we actually get this iron hydroxide. Sometimes we say iron 3 hydroxide to emphasize there is a +3 charge on iron. And then we need to dissolve it. We dissolve it with a strong acid, nitric acid, to form this iron 3+ again in an aqueous solution. So now this is actually soluble in the solution, and we use again a spot test to determine if iron 3 plus is indeed present in the initial mixture. So we're going to talk about the spot tests. First, let's just look at the identification of iron plus again. So after we add HCl in the unknown solution, we get some precipitate, but this precipitate can be silver chloride, it can be lead chloride. This is because both silver and lead may react with chloride to form white precipitate. So we need to heat it up. Again, I mean, after we heat it up, if you still see white precipitate, then you do know there's silver plus in the original mixture or unknown sample. And then we need to separate this precipitate from this liquid layer using a technique. It's called centrifuge. And we have supernatant on top of precipitate, and then we need to test if lead is present in the liquid layer or so-called supernatant. How do we do that? Again, we use a so-called spot test. We can react with the liquid layer, for example, the liquid layer on top of the silver chloride precipitate, and we react that with either these 5 reagents. All right, the first one is potassium chromate, and this is potassium thiocyanate and sulfuric acid, hydrochloric acid. This is ammonium hydroxide. We react this supernatant with all 5 reagents and record the observations. So you have 5 observations here. If there's no precipitate, just write no precipitate. If you see white precipitate, just say white precipitate. And not only that, because this is a spot test to identify the presence and identity of unknown cation, so we need to react some known cations with these 5 reagents. For example, we react this known lead ion with the 5 reagents and record the observations. Iron 3 plus with the 5 reagents, silver plus with the 5 reagents, and barium 2 plus with the 5 reagents. And we need to record all those observations. I'll give you some example in this table. Over here, when you react lead 2 plus with this potassium chromate, you're going to see yellow precipitate. So just write yellow PPT over there. But when you react this iron 3 plus with sulfuric acid, there's no precipitate, there's no apparent reaction. Just write no precipitate here. If you mix silver nitrate with HCl, you will see white precipitate. And if you pay attention to these 4 known chemicals, they contain lead ions, iron, silver plus, barium. But if you look at the anions, they are all nitrate, 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 nitrate. This is because the nitrate ion NO3 minus is always a spectator ion that does not react with any other ions. That's why we can just focus on, these observations and identify the presence of the cations. All right. So again, we need to fill out this 20 blanks, and then we need to do the spot test for a unknown cation. So if these 5 blanks, for example, match with these 5 blanks, and we know there is lead ion in the initial unknown sample. In this experiment, you need to write out the balanced reactions. Again, I'll give you some examples over here. First, the reaction between lead nitrate and potassium chromate. This is called a double replacement reaction. So over here, we have lead reacting with this chromate to form this precipitate. This is lead chromate precipitate with a yellow color, and potassium is combined with nitrate. We have potassium nitrate over here. We need to specify aqueous because they are soluble, and this is insoluble in water, so we put a solid here. The second reaction is more difficult because iron in this case has a 3+ charge, so we need to balance the charge carefully. We need to put a 2 here and 3 here so that in the end we form this and this. Let's examine this formula. We have iron 3+, and we have 2 of those. Therefore, we have a +3 charge on the cations. Sulfate has a -2 charge, and we have 3 sulfate, so in total we have -6 charge on the anions. In this formula, we have plus charge, +6 charge here, and -6 charge here. They are balanced. The third example is pretty simple. It's just silver nitrate react with hydrochloric acid, and then we form silver chloride, which is a precipitate. So you write solid here, plus this is nitric acid. This is aqueous solution, aqueous solution, aqueous solution. This one is precipitate, so we write solid over here. Safety. Whenever you use acids and bases, especially concentrated ones, be very careful. Wear gloves. If there's any spill on your skin, rinse it for 15 minutes. I have videos for all those chemistry 183 labs over here, so if you just click this playlist, you can see a bunch of videos on, for example, the pre-lab assignments, standard operations, and data analysis. And over here, this video tells you how to use a centrifuge, so I'll just click this and bring you to that video.
What if you could do SEO smarter and faster? With Wix, you can edit your meta tag.
Centrifuges can be used to separate immiscible liquids into layers based on density.
This video demonstrates the proper use of a centrifuge.
Pour the liquid you wish to separate into a test tube as another test tube placed directly opposite it.
Make note of the position number. Make sure every tube in the machine has a corresponding tube directly opposite to it for balance.
Alright, for the copyright of the video, I can only show you several seconds of this.